antimicrobial resistance, and we are looking at the legacy of IMI COMBACTE project. I am Nathalie Signoret. I'm a Senior Scientific Project Manager within the Innovative Health Initiative, and I'm very pleased to moderate this discussion of today. Before I introduce you to our distinguished speakers, I would like highlighting that the purpose of this event is to look at what are the challenges in the field of AMR that our project try to solve, what they have delivered in terms of results, and more importantly, the impact of these results that they have in our fight on AMR. And if we move to the next slide, you will see that for some time ago, AMRs and resulting infection in multi-drug resistant bacteria have been recognized as a major health threat in Europe and at global level. Back in 2012, the Innovative Medicine Initiative, which was our previous public-private partnership, launched a very large public-private collaboration program called New Drug for Bad Bugs, as one of the response to the 2011 European Commission Action Plan against the rising threats from AMR. Now, the program tried to look at different challenges. The science to get the antibiotic entered into the bacteria, a platform to boost the development of potential new candidates, the clinical development of new antibacterial treatment, and the economic model and stewardship on the use of antibiotics. Now today, and this is underlined in this slide, we will focus on the third challenge, meaning the clinical development on new antibacterial treatment. And our speaker will present you the major achievements from the COMBACTE project, meaning COMBACTE-NET, COMBACTE-CARE, COMBACTE-MAGNET. Now, of course, this project have done a lot of work and have delivered a lot. So this webinar will be only a snapshot of really the main results, but there will be more. And of course, we will invite you after the event to look at their website to see even further results that have been delivered by the project. But now it's the time for me to introduce to our speakers. And if we move to the next slide, I will go by the order of their intervention. We will start by Marc Bonton from the UMC Utrecht. Then we will move to the presentation of Jesus Rodriguez Baño from the Hospital Universitario Bergen-Macarena. Sorry if my Spanish is not that fluent. And then we will have the presentation from Bruno François from the CHU Limoges, and Halle Rogers from Pfizer. And finishing with some words from Hassan Jeffrey from Aridis Pharmaceuticals. But now I think it's maybe before saying, we will go through the presentation, and then at the end, you will have plenty of time for asking the question. To do the question, you just need to put your question in the chat that you have at your hand. And then we will go together with my my colleagues from the communication team. We will go through the question and we will address them at the end of the event. As I said, we will have plenty of time to go through. Now, be aware that the session is recording, and of course, the recording will be available afterwards, so you can always go back to it if you didn't follow entirely this session. But on this note, I think it's time to start, and now I will ask you, Marc, to take us through the presentation and the building of COMBACTE networks. Marc, the floor is yours. Thank you.
Thank you, Nathalie, and welcome to this webinar. And it's a pleasure for me to present what has been achieved over the last 12 years in COMBACTE. Next slide. So when we all started, it was recognized that antimicrobial resistance is, let's say, indeed the perfect storm. Infections caused by resistant bacteria are associated with worse outcomes and higher costs, and that jeopardizes modern medicine, for example, stem cell transplantations, ICU care, and major surgery. Antibiotic resistance, as you know, is increasing worldwide, and there is very slow development of new antibiotics. So the prospect of a post-antibiotic era is real. Next slide. So the objectives of COMBACTE when we started in 2012 was to increase the efficiency of antibiotic development, to align clinical trials with cutting-edge molecular methodologies and trial design. And in addition, to create a self-sustaining premier antibacterial development network by expanding research and laboratory networks, by optimally aligning clinical trials with investigator sites in Europe, and by delivering clinical and epidemiological data to support antibiotic stewardship. Next slide. Now, what makes studies in antibiotic resistance inefficient, as was mentioned by Nathalie and is widely recognized? There's a set of criteria, a set of reasons. One are the EARL hurdles, and EARL stands for ethical, administrative, regulatory, and legal hurdles that need to be, let's say, dealt with in each new study. Then there is a lack of appropriate expertise and training for executing studies in acute infectious diseases. And it's very important to realize that these are acute infectious diseases. So mostly decisions about enrollment of patients need to be made within hours, which is very challenging and also needs a microbiological diagnosis. If you look at the expertise and training, at that time there was an inverse relationship between the capacity to do such difficult studies and the prevalence of antibiotic resistance. Capacity was present in those countries with the lowest levels of antibiotic resistance. I also already mentioned the need of laboratory support for such studies, and there was a lack of laboratory support for such studies. There also was incomplete understanding of antibiotic resistance epidemiology. We know that resistance is different in different parts of Europe, but in the individual countries, it's still unknown where the highest prevalence of antibiotic resistance is. And finally, there was a lack of innovation in clinical trial methodology. Next slide. So to address these issues, we decided to build 4 networks, the network of networks that we created in COMBACTE: CLINET, LabNet, STATNet, and EpiNet. CLINET is the network of study sites, mostly hospitals, and the final phase is also primary care sites. The LabNet is the network of laboratories to support the clinical studies performed in CLINET. So LabNet and CLINET are very well connected. Then we have STATNet, and that's a network of experts in methodological and statistical aspects of clinical studies in infectious diseases. And finally, we established EpiNet as a network of experts in epidemiological studies in infectious diseases, in particular in antibiotic resistance. Next slide. If we briefly go over the achievements of these different networks, if you look at CLINET and LabNet, they have some shared achievements, and that was developing good clinical and good laboratory practices trainings, both in face-to-face meetings and online trainings. And in total, more than 1,000 physicians across Europe have been GCP trained. They also established protocolized site selection procedure. I will show more in the next slide. And during the projects, they continuously worked on the growing of the network. Then for CLINET, special achievements were realized for more efficient contracting of study sites, more efficient dealing with regulatory approvals of studies, better site management during studies, and building a portfolio of quality parameters for study sites that would help us in future studies with selecting the best sites. LabNet established good quality control systems for sites that wanted to participate in studies, dealt with the sampling, transports, and analysis of samples as a central laboratory facility, and created a biobanking and repository facility in Antwerp, our central lab. Next slide. Here we see some figures of what has been achieved in CLINET and LabNet. If we first look at the left side of the slide, we see the total number of hospitals in CLINET is about 1,200 and about 900 laboratories that are connected to the networks that are present in 42 countries, including all European countries. 1,455 laboratory contacts have been established, as with more than 4,000 hospital contacts. In total, more than 9,000 strains were collected, 23,000 microbanks, almost 100,000 samples were transported, and more than 100,000 specimens were collected in all the studies that were performed. On the right side of this figure, we see the composition of the COMBACTE networks over the 3 projects, 55 academic partners, 9 FPR partners, and as I already mentioned, 1,100 GCP, Good Clinical Practice certificates were produced. In total, 47 studies were performed, of which 27 were interventional studies and 20 were observational studies. 10 were in phase 2 11 in phase 3 and 8 phase 4 studies. We also used the network as it was available during the COVID crisis, and in the network, 9 COVID-19 clinical trials have been started. In total, more than 47,000 patients were recruited and 150 scientific papers were produced. The types of infections and the bacteria and viruses involved are listed here as well. Next slide. This is the site selection process that was established at the beginning of the project, and that was improved and used throughout the project and is still used. So it's a very transparent and objective way of selecting the best site for a specific study using all the available information that we have for such a site. It is needed to be transparent as we're using public funding, and we need to be able to justify why certain sites are selected and others are not selected for a specific study. So this site selection process is established for each new study in collaboration with the sponsor of that study and includes all the information about a certain study site in Europe during all preceding activities in our network. So in time, we have collected very much information about study performance and study capabilities of clinical sites and laboratories across Europe. Next slide. If we move to the next network, it's the APNet. And the achievements here are optimized surveillance for antibiotic resistance and infections in healthcare settings. It contributed to homogenization of current and future surveillance in Europe. It provided information for public health action, and it informed and guided R&D for new antibiotics for multi-drug resistant pathogens. In the next few slides, some summaries of the output. Next slide. Here we see a list of white papers on bridging the gaps between surveillance data and antibiotic policy and stewardship in hospitals, in long-term care facilities, in the animal sector, and in the outpatient sector. APNet also contributed to consensus and recommendations to support the homogenization of surveillance strategies across EU countries. So this was about the surveillance. Next slide. On its website, that you can see here, APNet has, let's say, regular updates of all kinds of epidemiological information that can be very helpful in designing studies and doing other aspects related to antibiotic resistance, such as the prevalence and incidence of antibiotic resistance in European countries, healthcare-associated infections in different countries, emerging resistances to new antibiotics, history of antibiotic resistance outbreaks, antifungal resistance, and it also lists the number of COMBACTE studies and the level of activity of these trials. Next slide. The final part that's shown about APNet is the central data repository that was created of 25 target pathogens and 5 targeted healthcare-associated infections with a bunch of information with more than 23 million strains collected, at least the information of these strains, and also strains related to infections. Next slide. The fourth network was STAT-NET, and the major achievements of that network were the application of population PK models for PK variability and optimized dosing, rank-based composite endpoints to improve power and relevancy in clinical trials, multi-state models to examine a range of time-dependent clinical outcomes, the use of historical clinical trial data for design and analysis, and teaching of the next generation of clinicians and researchers on platform trials. In this publication in Clinical Infectious Diseases, multiple aspects of the work of the COMBACTE STAT-NET group are presented. Next slide. Two important achievements were the work on the novel endpoints for ventilator-associated pneumonia in randomized controlled trials in a series of 3 publications on the evidence synthesis, the expert consensus, and the implementation of such new endpoints. And the group also produced a Shiny app, improving insights in competing time-dependent outcomes in randomized controlled trials, and that has been published recently in the reference that you can see here. Next slide. To end, all these efforts, all these networks have led to a very, let's say, what we think is a very good network to make trials and to perform trials more efficiently, and one of the goals was to make this sustainable. That has led to the creation of ECRADE, the European Clinical Research Alliance on Infectious Diseases. So at the end of the project, of the COMBACTE projects, we moved from a public-private partnership consortium to a not-for-profit foundation led by academic investigators under the name eCRATE. And the CLINET, LabNet, StatNet, and APNet are still the pillars of eCRATE as they were established in COMBACTE. eCRATE is led by an international leadership group. It's based in Utrecht, where its headquarters are, and has currently 80 employees for research support. It has a management board of 3 persons and 2 researchers, the leaders of CLINET and APNet, the coordinating committee of 17 academics from 11 EU countries, and the clinical network has a council of 24 network leaders in different countries, in 19 different EU countries. The current activity of the network is 11 international studies in 269 study sites in 24 EU countries, and all supported by a fully operational data collection tool according to all FAIR data principles. With that, I end the presentation on the networks and give the word back to Nathalie. Next slide.
Thank you very much, Mark, for this overview and showing the importance of the creation of the different networks that are definitely important when running clinical trials. You mentioned about epidemiology, and then I give the floor to Jesus to take us through observational studies in COMBACTE. Jesus, the floor is yours. And before you start, Jesus, just a reminder, please put your question in the chat as we will go through them at the end of the presentation. Thank you.
Thank you very much, Nathalie. Thank you very much for the invitation to participate in this webinar. Well, my deal is to try to discuss and explain the observational studies performing COMBACTE. Next slide, please. As I understand that many of you come from different fields of knowledge and different areas, you may not be familiar with what are observational studies, and as a difference with randomized controlled trials in which patients are randomized to receive one or other treatment. In this case, we just observe the reality. So there's no intervention in this type of studies. It's typically done to try to understand the burden of disease, the risk factor for acquiring an infection, in case, some aspect of the management of the infection, as physicians do when they face a patient with this type of infection, and which are the predictors for the outcomes of the patient. They are also typically very helpful to understand which are the priorities in research and in this area in antimicrobial resistant research, and which are the most important gaps in knowledge that can be answered by these or other types of studies. They are also can be also very useful to inform a more efficient design of randomized trials. As Mark explains, randomized trials may have some problems in their designs, and by knowing to which patients this trial should be targeting, we can do trials which are more efficient. And also we can provide additional and more, let's say, generalizable information about real-life data. We have to understand that the information coming from observational studies has some limitations to assess causality, particularly if we try to compare the efficacy of different treatments, because we are not randomizing, and therefore other variables can influence the outcome of the patient beyond the treatment. Next slide, please. So this is the list of the observational studies that were performed under the COMBACTE project and the 3 sub-projects like COMBACTE-NET, COMBACTE-CARE, COMBACTE-MAGNET. You can see there are a good number of projects as far as I anticipate, outreach, expect, honest, PrEP, Eureka, rescuing. All these projects were trying to provide that information that I explained before for the development of randomized trial, if possible, later. And they will deal with almost type of infections like ventilator-associated pneumonia, surgical site infection, prosthetic joint infections, and urinary tract infection. And also they were targeting specific antimicrobial resistant pathogen or pathogen that, even if they're not antimicrobial resistant, are very important and critical for this infection, like Staph aureus, Pseudomonas aeruginosa, Clostridium difficile, and the resistant carbapenem resistant Enterobacterales or Acinetobacter baumannii. All these studies have been published, and you can see the publications, and you can have access to the publications in the share code that you can see in the slide. Next slide, please. Just to provide a specific example of how this is done and how this was done in the COMBACTE project, I will take the example of carbapenem-resistant Enterobacteriales. This is one of the most critical problem in antimicrobial resistance, and it is listed in the top priorities from the WHO to develop new drugs and new research in antimicrobial resistant pathogens. So there were two important questions for this infection. The first was, what are the risk factors for acquiring one of these infections? And the other question was, what is the impact of this resistance in mortality? Why are these questions relevant for randomized trials? Well, with risk factors, as I explained before, we can improve the design of the randomized trial by targeting the trials to areas and type of patients in which this type of pathogens will be more frequent. And also, the impact on mortality is important in order to understand what sample size is needed in this randomized trial, because we can understand what is the opportunity to improve that outcome with new drugs or with new treatments. Of course, there are other academic or, let's say, important information that we can get from these studies, but I will just summarize the information for these two specific aspects that are relevant for randomized trial. So for this, we developed a design. We designed a study as a multinational match case control study. So we compare what we call a case. A case is a patient with an infection caused by a carbapenem-resistant Enterobacteriales infection, and we compare them with two types of control patients. One are patients infected with a carbapenem-susceptible Enterobacteriales infection, which is CSE in this slide. And also we compare patients with the carbapenem-resistant infection with patients without any infection in their patient. We match them by other variables. And as you can see in this study, we collected information from 235 patients with CRE infection, also 235 patients with CSE infection, and 705 patients without infection. And we, as I explained, match them by other different type of variables. Next slide, please. So by having this design, we could understand which are the risk factors for this type of infections. And as you can see here, what you can see in green are specific variables that were significantly associated with an increased risk of having an infection caused by a carbapenem-resistant Enterobacteriaceae, both if we compare them with carbapenem-susceptible or if we compare them with with non-infected patients. You can see that some variables were common for the two type of controls, like previous colonization by a carbapenem-resistant Enterobacteriales, or having a urinary catheter, or having received previously antibiotics in the last 3 months. And there are other features that are more specific depending on the type of control that we are considering. So by this type of information, we can inform that if we want to make a trial, we can, if we want to find patients with this type of infection, we should probably try to target this patient population in order to have a very efficient recruitment for a randomized trial. Next slide, please. And continuing with our second question of interest, which is what is the attributable mortality for this infection, you can see that comparing our case patient with carbapenem-resistant Enterobacteriales with the other two type of control patients, in the left part of the slide, you can see the overall mortality, and you can see that mortality in patients with CRE was 23% compared to 10% in patients with carbapenem-susceptible Enterobacteriales, or 8% in patients without this type of infection, meaning that there is an increase in mortality that is around 13%. If we can also divide this type of patients according to the specific type of infection, we have patients with complicated UTI, this complicated urinary tract infection. We have patients with pneumonia, we have patients with complicated intra-abdominal infection, or we have patients with whatever the infection, but additionally having a bloodstream infection, so the bacteria can be isolated in their blood. You can also see in the slide the difference in mortality. In UTI, the difference in mortality is around 10%. In patients with pneumonia, it's around 16%. The difference is less significant in patients with complicated intra-abdominal infection, and the difference is even higher in patients with bloodstream infection. So this information provides us data about how important these pathogens are. They increase mortality by themselves, and also how much we could reduce this mortality by having a new drug that is fully effective against this bacteria. Next slide, please. And just to finish my presentation, I would like to comment on what are the added value of these observational studies that we perform in COMBACTE. As Mark explained, we were able to use a clinical and laboratory network to run very complex multinational studies across Europe. The sites that were participating in these studies were trained to include patients in the observational studies, and that provided them also with training in case they could participate in a randomized trial, because they were already trained to detect these patients, to collect the data, and so on. And also, we were able to perform complex data analysis with the help of COMBACTE-NET, as also Max explained. But also, the data from these patients are, the aggregated data are incorporated to the EpiNet repository, providing a knowledge that can be applied in other fields, and also the possibility of data sharing according to the requirement for data sharing in this type of studies. And of course, these preliminary data of these studies were used for the design of randomized trials that were performed during the COMBACTE project, having a target rich population, helping to calculate sample sizes and other aspects of randomized trial. Next slide. And I think that's all. Thank you very much.
Thank you very much, Jesus, for taking us through the epidemiology and the importance of it and the work that was done in COMBACTE. Again, I would like to remind everybody that if you would like to ask a question to Jesus in particular, or to all the speakers, please use the Q&A box for the question. So that's the function where you can plug your question, and we will go through at the end of this event. So now that we had the presentation on the epidemiology and the importance of it to help with the design of a randomized clinical trial, let's move to some example of a randomized clinical trial. And Halle, I leave you the floor to take us through. Thank you.
Thank you very much, and thank you for the opportunity to present today. My name is Halle Rogers. I'm representing Pfizer. We're going to talk more about the journey of the IMI impact on AMR. Next slide. So, as stated on the COMBACTE-CARE site, the project overall aims to, number one, better understand multi-drug resistant bacterial infections such as carbapenem-resistant Enterobacterales, which were introduced to you by my colleague Jesus, and to support the development of new treatment options. Number two, analyze observational, clinical, and epidemiological data sets to inform the design of randomized trials. And finally, number three, make significant contributions to the development of a novel treatment option, Astrionam avibactam, or ATM-AVI, for patients with serious gram-negative bacterial infections, including those caused by a particular type of carbapenem resistance called metallobilactamases, for which there are limited or no treatment options. And this is where we will focus my presentation today, building on the excellent work that my colleagues Mark and Jesus already introduced you to. Next slide. So you've heard a little bit about antimicrobial resistance today, and we know it's a growing problem worldwide. Infections caused by bacteria known as these CREs are resistant to most available antibiotics and are so difficult to treat. They are considered to be one of the most dangerous drug-resistant bacteria in the world. Cases of CRE infections are on the rise in Europe and globally, and we know this because of some of the important epidemiological work that's been done, as been shared by my colleagues. And so you can see on the right-hand side of the slide throughout Europe, there's evidence of this resistance. And so we see anywhere from no cases reported all the way to an endemic situation in some countries. Next slide. And so where our phase of research comes in is through the conduct of clinical trials with humans. And so our phase 2A study was called Rejuvenate. It was a prospective, open label, multi-center, non-comparative dose selection study. So a lot of words that essentially mean we conducted it at multiple centers, 40 to at multiple centers across Europe. And we were trying to find how the medicine, ATM-AVI, interacts with the body. Is it safe and tolerable? And does it work? And at what dose? And so what we did is we recruited adult patients between the ages of 18 to 90, and they must have had or were planning to have a surgery within 24 hours of administration of the first dose. And they must have had a diagnosis of complicated intra-abdominal infection and must have been hospitalized. And so we started with cohort 1, which was a small group of 16 participants with normal renal function or mild impairment. And then we escalated at a different dose up to cohorts 2 and 3, which you see on the right slide. In the Rejuvenate study, COMBACTE-CARE enrolled all 40 patients across 3 countries in Spain, France, and Germany. Next slide. Now, as we did with the observational studies and the work that Mark described, we built on our lessons from the first Work Project 2A Rejuvenate study, and we're able to translate that into the larger Work Package 2B Revisit study. So COMBACTE-CARE Spanish site were particularly successful at recruiting patients for the Rejuvenate study. This was in Seville. Their learnings were key to support recruitment initiatives through the creation of a video highlighting the importance of a multidisciplinary team and coordinating communications for successful recruitment and retention. These lessons allow the network to build upon the efficient and effective clinical recruitment within COMBACT sites and translate them into the Revisit study, just as we had done before with each subsequent phase of the entire program. Next slide. The Revisit study was much larger. So rather than less than 50 patients, we're now looking at a sample size of 422 participants. This was a phase 3 prospective, open label, multi-center comparative study. And we had participants who either had a diagnosis of complicated intra-abdominal infection or pneumonia, and that was either hospital-acquired or ventilator-associated pneumonia. And they must have not only been in the hospital, but must have required IV antibiotic treatment. Upon entry, they were randomized to one of 2 arms, either the experimental arm, which was ATM-AVI plus or minus metronidazole, or an active comparator arm, which consisted of meropenem plus or minus colistin. Our primary endpoint, so what we were looking to achieve as part of this research, was what we defined as clinical cure at day 28 in the population that was enrolled. This study was conducted in approximately 165 sites across 20 countries in the America, Europe, and Asia. So you can start to appreciate that the groundwork that was laid before the conduct of this trial with creating a network of sites, understanding where the problem was, is absolutely what contributed to our capacity and our ability to conduct a study that was so large. Next slide. So approximately 50% of all subjects randomized into the Revisit study were randomized at COMBACT-CARE sites, making COMBACT-CARE a key contributor to the study's success. 4 of the top 6 recruiting sites around the world were within the COMBACT-CARE network. And you can see on this slide, I've plotted the study site locations. And if we were to refer back to that original plot that showed where CRE infections are growing or are already endemic, you would see a very clear correlation. Next slide. We faced a number of challenges and strategies despite all of the pre-work, some which we could have anticipated and some certainly which were a little bit of a surprise to us. Starting at the very top of the list on the left, which was the outbreak of COVID-19. So Revisit was conducted between the years of 2018 and 2023. And so we faced a number of challenges during that time. In addition to COVID-19, there was geopolitical conflicts, site dropout, loss of motivation. There were recruitment challenges. We encountered clinical drug supply shortages, lab supply shortages and shutdowns due to the pandemic. There were importation restrictions that were specific to different countries. And as you could imagine, there were changes in standard of care since we started the study in 2018. But what we were able to do in collaboration with our COMBACTE-CARE colleagues is to develop strategies to address each of these challenges. So number one, we maximize site and country footprints. So you saw we had 165 sites around the world. Patience and compassion for their sites and their priorities. We embraced collaborative, proactive, and targeted site selection. We optimized manufacturers and suppliers. We implemented just-in-time training and training tools, so training at the time that the sites and their staff needed it. And finally, we worked closely with regional contacts and leveraged their expertise to how to address some of these issues at a local level. Next slide. We also developed what I call revisit-specific approaches. So these were approaches that we learned through earlier phases, not only of the clinical trials, but also of the collaboration as a whole. So we implemented and emphasized collaborative interdepartmental hospital study teams. As mentioned before, we used an enrollment video to help to train and educate sites on how to have that collaborative study team. We implemented what we call a patient pathway identification exercise. So we knew that these patients were presently in the hospital, but how did they get there and how are they identified by the hospital system to be enrolled into a clinical trial? We conducted virtual investigator meetings and remote monitoring as permissible by local regulations. We had to allow for clinical drug supply flexibility, so we contracted with multiple drug supply manufacturers as some ran out of supply or were unwilling to distribute. Our academic leads implemented a 24/7 telephone investigator support line. We developed study-specific recruitment support tools in collaboration with our colleagues in Tel Aviv and support training videos. And finally, our academic lead role in oversight of planning, study conduct, and medical support was our last piece of the puzzle. And I want to emphasize here the collaboration across the COMBACTE-CARE partners was key to the success in not only starting and designing, but completing the Revisit study. Next slide. What I think is important here is to understand what the impact of all this work was. The COMBACTE-CARE demonstrated that and how a public-private partnership between industry and academia can work, reinforcing our common goal in finding novel solutions for patients with serious infections. These clinical studies that I've just shared with you have led to an approval of a new antibiotic, and you can learn more about it on the Imblavio European Medicines Agency website link there. The close relationship grown from this consortium, I think, will help the global fight against AMR, antimicrobial resistance, and provide evidence that Europe has the capacity and infrastructure to conduct clinical trials for novel antibacterial agents. Thank you very much.
Thank you very much, Halle, for taking us not only the challenges, but as well the great work that was done in conducting this randomized clinical trial and what it led to a new antibiotic on the market, or at least available in Europe now. Again, as a reminder, please put your question in the Q&A box at the bottom of your screen so that we can take them through once we have the Q&A session. But now let's hear from Bruno to take us from the COMBACTE clinical trials from the investigator's perspective. Bruno, the floor is yours.
Thank you, Nathalie. So I'm Bruno François. I'm a critical care physician working at Limoges, France. I'm an investigator. I've been working in clinical research for now more than 25 years. I spent at least 2/3 of my career running clinical studies. So I have a little bit of expertise in this field, and COMBACTE has probably changed a little bit the model, and I will try to convince you about that. So you have seen the pharmaceutical company perspective with our colleague from Pfizer, and I will show the other part, which is the investigator perspective. Next slide. So you have learned a lot about the networks, and I will give you some example of what we did in the different consortium, COMBACTE-NET, COMBACTE-CARE, COMBACTE-MAGNET, and COMBACTE-CDI. A lot has been achieved, as mentioned by Mark, and maybe I will spend a little more time on maybe what were the most innovative trial that we have run in COMBACTE. So first, next slide, please. So the first one is Aspire ICU, and this has been mentioned already by Jesus. So before designing clinical trial, especially in early phase, even if the concept are pretty strong, you need some strong data. Of course, you can go to the literature, but sometimes you don't have an appropriate answer, and you need some more robust data to strengthen your design. And so Aspire ICU is dealing with VAP. So for those of you who are not familiar with ICU and VAP, when you are intubated and receiving mechanical ventilation in the ICU, you have what we call colonization into the lung, which means that you have bacteria that are documented into the lung, but you are not yet infected. But this colonization can then move to a true infection, which is called VAP, ventilator-associated pneumonia. And this is caused by virulence factors from different bacteria. The two most important bacteria in VAP worldwide are Staphylococcus aureus and Pseudomonas aeruginosa. And even if we knew already a lot on VAP and a lot about Staphylococcus aureus, we were missing some information on the true epidemiology, and this is what has been done. It was led by UMCU, Fleur Palin in the Netherlands, and 2,000 patients has been included, which is probably the biggest observational study done in the ICU. Next slide. And this gave us really robust data to design satellite, the satellite program, which is divided in two parts, satellite 1 and satellite 2. Satellite 1 was the phase 2, and satellite 2 was the phase 3. It is probably one of the most innovative trials that has been done in VAP using monoclonal. Monoclonal is a non-antibiotic drug. Instead of killing the bacteria, the monoclonal will target will link with a specific target. And in satellite, we were using a monoclonal, which name was suvratoxumab. And suvratoxumab act against alpha toxin. Alpha toxin, which is also called alpha hemolysin, is the toxin which is the most important virulence factor for satellite, for staphylococcal infection. So in fact, when you are colonized with Staphylococcus aureus, because of alpha toxin production, you have a sort of damage of the lungs that will create infection and what we call staphylococcal VAP. And so the innovation of this trial, instead of treating the event, the VAP, it was what we call a preemptive approach. So we will select a patient being colonized, colonization being documented with PCR, in a real-time fashion, which was another innovation of this phase 2. And then when patients were documented with Staphylococcus aureus, we were giving them a single shot of monoclonal. And one of the interests of monoclonal when compared to antibiotics is their duration of action, which can cover several months. And the intent and the objective of Satellite 1 was to demonstrate that we were able to reduce the number of VAP event in patient colonized with staph, and we have been able to achieve that. It was a huge trial, 800 patients screened, which is a lot for a phase 2 trial, with 200 patients being randomized in 9 countries all over Europe. So it was a European trial. So thanks to promising results, this trial has now moved to a phase 3, which is Satellite 2, which is exactly the same design. Next slide. Evade is dealing with Pseudomonas aeruginosa. Pseudomonas aeruginosa is the other bad bugs in the ICU, which is the other important pathogen driving for VAP in a ventilated patient. And we, thanks to what we have achieved in Satellite, we have designed more or less the same type of trial, documenting the Pseudomonas colonization with PCR in a real-time fashion, and using another monoclonal, which is Gremubamab, which is what we call a B-specific monoclonal. Instead of targeting alpha toxin, there were 2 targets, PCRV and PSL. Those are 2 important virulence factors of Pseudomonas aeruginosa, and so the objective were more or less the same: selecting a patient being colonized with the Pseudomonas, and then giving a single shot of monoclonal to see if it may reduce the number of VAP events. A lot of screening, 1,000 screening, roughly 200 patients being randomized, same number of sites, same number of countries than in Satellite. Unfortunately, the results were less promising, even if we have generated some interesting data. But probably the picture is more complex with Pseudomonas than with staph. And next slide. Last trial to be mentioned, which is the Honest Prep trial, which is observational, which is not an interventional, but with the same intent that we did with Aspire ICU, and keeping the experience and what we have learned on VAP. The idea is to set up a sort of VAP network all over Europe, and the intent of Honest Prep was to generate some prospective and robust epidemiological data on VAP, to be able to design more appropriately VAP trial in the coming years in a CRED, as mentioned by Mark before. Next slide. So all this trial has been published in the very high impact factor journals, such as Critical Care or the Lancet Infectious Disease. Next slide. So the story was completely different than a classical partnership between a pharmaceutical company and an investigator. As mentioned before, I've been working with pharmaceutical company for years now, but the level of involvement was completely different since the very beginning, not only with the design. As an example, in Satellite, we were starting almost from scratch, so we had to define new definition, completely innovative design with a preemptive design with a monoclonal, documented patient using a PCR tool in a timely fashion. So being able to design such a complex trial would probably not have been possible outside COMBACTE, in which usually the collaboration between academia and pharmaceutical company is less strong. So all the discussion were highly transparent, because we are within a non-competitive model, non-commercial model. The communication is highly direct. As I always say, I spend probably more time with Hassan Jaffrey during the entire COMBACTE story than with my wife. So really close collaboration with the company, with interdisciplinary collaboration. And there were a really deep involvement of partners at every step of the trial, not only in study board, that is being pretty classic, but also in site selection in collaboration with the different network mentioned by Mark, CleanNet and LabNet, with equal voting member, which is not a classical picture. Usually the last say is on the pharma company side. We were also able to discuss with regular bodies, EMA and FDA, which is not highly classical. And we are also access to the entire data set, and the publication was done collaboratively between all the company and the academic. And last but not least, we had also access to all the biological sample. One part of the objective was also to create biocollection and really innovative biocollection in some very specific topics. Next slide. Also some networking activity between different teams. It has been mentioned by Mark before, but thanks to CleanNet, now we have a pretty strong network all over Europe in different countries, creating a collaborative and collective dynamics in clinical research that had benefited from one trial to the other. Acquisition of new skills, capability building, thanks to some specific training, especially when using innovative design, but also, and this has to be highlighted, some opportunity for less experienced site personnel that were given the possibility within the consortium to visit and to learn from a top performing site. Next slide. What for the patient? First is to benefit to possibly from new innovative drug, and monoclonal antibodies is probably highly innovative and with probably a much safer profile, safer safety profile than antibiotics, as an example. But also having academic on board, paying more attention maybe to patient than in a classical collaboration. This gives some more realistic and feasible design, especially in some specific difficult settings such as the ICU. Next slide. And this will be probably my last slide, as mentioned by my other colleague. I think one of the greatest achievements is the creation of international and investigational networks with, at the country level, what has been called National Clinical Liaison Team. Now, CleanNet LabNet covers more than 400 countries. So this helped to set up and to start new trials, and what has been learned, especially in VAP, will probably benefit to some other companies or some other academics. And now all what has been generated in COMBACTE is now integrated into a CRED. And with that, I will thank you again for the invitation.
Thank you very much, Bruno, and thank you for highlighting the difficulty of some of this trial, but at the same time, what will remain after the COMBACTE project. Before we move to the Q&A session, and again, please use the chat or the Q&A box to ask your questions. Hassan, I will just ask you to take on some of the message on some of the your experience having been in this project and some take-home message.
Thank you, Nathalie. So hello, everyone. My name is Hassan Jaffrey. I'm an infectious disease doctor. I was previously an academic physician and researcher but now I have been developing novel anti-infectives for over 25 years, first as an academic researcher and more recently at industry. In addition to being the industry lead for 2 monoclonal antibody programs, I have also served as the coordinator for COMBACTE-NET and COMBACTE-MAGNET consortia. So really had the pleasure of following the growth and development of COMBACTE through its inception. One of the substantial long-standing gaps in infectious disease research have been the absence of clinical trial networks, including researchers as well as microbiology laboratories, especially in the area of antibacterial development. Similar networks have long existed in other specialties such as oncology, autoimmune diseases, neurosciences, and HIV/AIDS, and have provided a lot of benefit and efficiency in developing novel medicines in those areas. This lack of such infrastructure in infectious disease has caused inefficiency and high cost. Historically, individual infrastructure would be developed at great expense and time for each clinical trial, and then would be dismantled after the study was completed. And this would lead to, in addition to expense, a great loss of knowledge that was created, and then people would have to reinvent that wheel again. So COMBACTE has been a story of a successful public-private partnership due to, I believe, a true collaboration and complementary relationship between all key stakeholders. In this case, especially the academic, physicians, researchers, and the pharmaceutical industry. The academic partners represent the leading clinicians and clinical researchers with intimate understanding of disease and unmet medical need for their patients. They understand what is needed for their patients to get better, what type of medicines are needed for those patients, and should be prioritized. And then these academics also include some of the premier preclinical and translational scientists, as well as some of the leading epidemiologists. I also feel that a consortium structure also allows for a scientific alignment and shared vision among its members, as opposed to the traditional sponsored clinical trials. On the other hand, pharmaceutical industry partners bring a track record of drug discovery and manufacturing and conducting large-scale global clinical trials and having extensive regulatory expertise. It was this shared leadership and accountability and the ability to bring all of these stakeholders with different areas of expertise, which was what made COMBACTE a successful public-private consortium. COMBACTE conducted multiple large-scale epidemiology and interventional studies aimed at changing the paradigm of research as well as clinical care, as has described by Dr. Mark Monton, Dr. Jesus Rodriguez-Spano, and Haley, as well as Bruno Francois. And there are several examples. It conducted research in novel molecules that were active against antimicrobial resistance, AMR, both antibiotics such as ATM avibactam, that was the development of which was described and shared with you by Haley Rogers, as well as non-antibiotics such as monoclonal antibodies, which were described by Dr. Bruno Francois. COMBACTE, in particular, actually took a lot of initiatives in designing superiority studies, which are actually quite unusual and novel in the area of antibacterials, where, and by virtue of superiority design, these studies are designed to really allow the clinicians as well as the public to identify the greatest benefit from any new medicine being studied. Novel clinical endpoints were also developed in collaboration between all the different stakeholders, especially including some of the leading pneumonia and ICU experts, which were received quite favorably by the European Medicines Agency and the FDA. COMBACTE has focused on developing a clinical network, CLINNET, and microbiology laboratory network, LabNet, in addition to its epidemiology network, the EpiNet, which I would encourage you all to actually visit the website, because it actually allows anyone in the public domain to really enter the website and have access to all the antimicrobial resistance data that's being collected on a daily basis through the member hospitals. And it's really an immense clinical as well as research tool. And then StatNet, which was responsible for creating and identifying novel trial design methodologies. Close to 1,000 centers have received GCP training, and a large number of microbiology laboratories have received laboratory trainings. These initiatives have not only helped enhance the research infrastructure within COMBACTE, but it has also led to a substantial improvement in level of patient care. This became quite evident during the COVID-19 pandemic. As the coordinator of COMBACTE, I had the opportunity to visit well over 100 academic centers and hospitals and microbiology laboratories throughout Europe. And many of these places mentioned that they had received either laboratory training or molecular diagnostic equipment through COMBACTE, which was being utilized now during the pandemic by those institutions for testing for COVID. So this is just an example of when you actually invest into developing and enhancing the infrastructure for clinical research, you will actually see a lot of other benefits as well. So the ND4BB, or the New Drugs for Bad Bugs program in general, and COMBACTE Consortium in particular, has been a truly transparent collaboration, as has described in detail by Bruno Francois, and has established itself as the new model for effective antimicrobial resistance drug development, leveraging the collective and complementary strengths of all of the key stakeholders. COMBACTE has also made studies more efficient through the research infrastructure it has created, which is now being offered within the new academic network, eCRATE. And one would hope that industry would, as well as other stakeholders, would utilize the eCRATE network to help develop novel antibacterial agents much more efficiently to help fill the needs of the area of antimicrobial resistance. So with that, I'd like to thank you all.
Thank you very much, Hassan. And I think all of you now have quite an understanding of the main achievement coming from the COMBACTE project, but more importantly, the sustainability and really what remains from these results and the impact that it will continue on our fight against AMR. So we said already that you can use this chat, or not the chat, sorry, the Q&A box in order to put your question. You have already received a couple of questions, so maybe I will start with the first one. And I don't really know who from you would like to answer, but maybe Jesus, because we try, if you can put back your camera as well so that everybody will be able to see you. So the first question we receive is, what regulatory hurdles are specifically meant and what should change? So I know that can be quite an extensive question, and I'm sure that all of you have some views, but let's try to focus. I don't know from you who would like to address it. Maybe we'll start with Hassan and Mark.
Thank you. Yeah, I can initially answer that from my perspective. I think the regulatory hurdles that one of the ones to perhaps use that as an example are new endpoints. COMBACTE had focused on what is known as preemptive treatment, as Bruno Francois described. So, which was basically the approach of trying to prevent an infection in the ICU in patients who were high risk and were on the ventilator, were not able to breathe on their own, and therefore they were considered to be at very high risk for developing pneumonia and lung infection in the ICU. There are plenty of trials that have been conducted for treatment of pneumonia, but there had not been conducted many trials for prevention of pneumonia by targeting a high-risk population. And because of that, there were no previously agreed upon endpoints, and there was limited guidance from FDA and EMA. And this is where the expertise of the academic experts, the clinicians such as Dr. Bruno Francois and Dr. Mark Ponton was critical, because they were able to, because of their understanding of their patient's disease, they were able to propose the endpoint that would be that would allow the researchers in the trial to have the best opportunity to evaluate whether the drug was effective or not in preventing pneumonia. So that endpoint of prevention of pneumonia, there was no previous regulatory precedent, but what was proposed, because from the experts, was actually widely welcomed by the regulatory agencies, and actually led to the first ever accepted definition of that endpoint. And that endpoint has subsequently been utilized by other clinical trials, a few of them outside the COMBACTE network. So that's an example of COMBACTE has created not only infrastructure, but also research tools to conduct some of these novel trials.
Thank you. Anyone wants to add something? I would ask you to keep the answer very short so that we can go many of them. Anyone wants to add on the regulatory challenge? I mean, we know that there are quite a lot in design, study design, on the, as you said, the endpoint. Anyone else? So let's move to the next question. Do you consider as a consortium implementing novel approaches to identify actually novel antimicrobial agents, such as machine learning, deep learning strategy? Who would like to take this one? Mark, Bruno, Jesus?
Maybe I can briefly comment on that. Well, I think that big data and machine learning are already being very helpful in developing some type of studies, and could help us to more rapidly perform some studies. But we should acknowledge that they're still not free of biases, and randomized trials have not been still probably will not be substituted by this. So these methods can help us to be more efficient, can help us to characterize our populations in a more efficient way, more rapidly, but and help us, of course, in our data analysis. But a randomized trial will still be needed. We need to probably, as Mark discussed, see how can we improve the design of randomized trials, and also discuss with regulatory bodies how to implement that in practice. So these are useful tools, but are not really changing the picture of randomized trials needed at this point.
Thank you very much. I guess if the question was about identifying, of course, the focus on the COMBACTE projects were more on the clinical development of already candidates or quite advanced candidates, rather than identifying potential candidates in order to be developed as a compound or new antibiotic. So if we move on to another question, would be like, so maybe that's a question to Bruno and Mark and Jesus. As an investigator, what would you say is the big advantage to be part of the network?
I can answer that. Can you hear me?
Yeah, we can hear you, please.
Well, being part of the network is that I think what we have established over the 12 years is now a very, I think, efficient network across Europe, where we can do trials much more efficient than we could 12 years ago. We have a much better understanding of the sites in Europe for certain trials, related to the local epidemiology of antibiotic resistance. The hurdles around ethical approval have also been reduced by the CTR that is now in place. So we were confronted before the CTR with ethical approvals in all individual countries, leading to huge heterogeneity. We've also built up huge experience in contracting sites and how to do that more efficient. And still there's a lot to win in that respect. And we have been working on innovating clinical trials, which became, let's say, very efficient during the pandemic for COVID, such as the adaptive platform trials. And I think the development over the next years will be to use these trials and to improve these trials in such a way that they also will become acceptable for regulatory purposes.
Thank you very much. And a second question, as if I was a new investigator, how can I join the network?
Well, the network is now ECRATE, the European Clinical Research Alliance on Infectious Diseases, and that's a very open network, so anyone that is interested to join can join. Look at our website, send us an email, and we have at this time point 11 studies ongoing, and in most of these studies, sites are welcome to join. And if you have ideas for individual studies to be performed in the network is open to investigator-initiated studies, but also to, let's say, studies initiated by the biotech companies or pharmaceutical companies to help them get their development plan executed.
Right, thank you very much. The next question, I think I will put it to Ale. How do trial costs for a phase 3 trial performed by COMBACTE relate to cost for a similar study performed by pharma? I don't know if you will have this granularity, but maybe if you don't have in terms of precise costs, is how much you have seen that the conduct of the phase 3 trial in COMBACTE has been in a way easier or more effective than being conducted outside. Ale?
It's very difficult to say. There is a huge spectrum of costs in the conduct of clinical trials. What I think is a value add for the COMBACTE network is that so much work was done leading up to the phase 3 clinical trials. So the observational epidemiological trials, they really helped ultimately to reduce costs in this model because number one, they told us where to go. So what countries, what investigators, they helped us to align with the investigators who would be able to recruit because that's typically one of the highest costs of any study is to bring sites on board that ultimately never recruit. So I would hypothesize that it was cost reducing, but I don't have the visibility just across the company as a whole to say, you know, this was less or more than a standard clinical trial. But I do think that there were substantial potential for cost savings initiatives because of the way the project was designed.
I can add to that we are, of course, analyzing all the data that we have on the study execution to see how our timelines have changed over the years in reducing the time to first patient in a trial and increasing the proportion of selected sites that really enroll during studies. Our ambition is to break the 80-20 rule so that 80% of the patients in a trial are enrolled by 20 sites, 20% of the sites. We want to have a much more equal distribution of all sites enrolling, and in that way achieve, let's say, the endpoint of the study faster.
And that indeed is very important because we see that's so often an issue that a lot of sites open that maybe do not recruit, or once you are recruited to do the retention. But maybe it's another question to you, Mark. How should the company best approach COMBACTE and now more ECRATE for evaluation of new antimicrobial?
So what we have established now is several what we call perpetual observational studies for relevant infections such as ventilator-associated pneumonia, complicated UTI. So that's a network of about 40 to 50 sites across Europe that use a master protocol for enrolling such patients without intervention yet. These sites are trained to identify the patients, to take the informed consent, to collect information, to have the same diagnostic approach, diagnostic criteria. And in such an environment, and we think it's new trials can be embedded very easily. So that would be, let's say, my recommendation. So if you are in that area, please contact us and we can see if we can help you with that development. For other studies to be developed in the future, we are, of course, more than willing to help in thinking about the design of the study and also the execution of the study. We have this network now up and rolling, but sometimes we get requests for very specific resistance problems for patients with those infections to be included in trials. And then we can easily, with all the information we have in our APNet, in our database on previous studies, go to countries and go to sites where those pathogens are prevalent in order to augment the enrollment of such patients.
Thank you very much. I think the next question will be probably again to you, Ale, and as well, Jesus. IHI is aiming at developing pre-competitive public-private partnership studies. The Pfizer phase 3 study is definitely a pivotal indeed. And maybe I should specify that indeed we are pre-competitive in the pre-competitive space. There was this exception on the OMR considering the importance and really the challenge and the threat. So these are the exceptional, in a way, project that we have been involved or we have been supporting to some extent the phase 3 clinical trial. But the question is, what are the specific criteria which differentiate a sponsored trial pivotal study from a study sponsored by COMBACTE? So I don't know if you would like to further address that, Ale, and
So, you know, the sponsor role is defined in a fairly specific way, and it has repercussions throughout the conduct of the study. So from my perspective, and I hope this is addressing the question that was raised, because we were collaborative sponsors, it allowed for input into study design at the output, not only at the study level, but also at the program level. And so while each entity had its particular responsibilities, so for example, Pfizer as a study sponsor was charged with conducting the study, but we collaborated with other entities to select sites and to educate the sites. And so the sponsorship role will translate into certain responsibilities in a given context. And that is not only important at the initiation or the design of the study, but also well through submission of the study results to various regulatory agencies. So there are actually, for Revisit in particular, we are partnering with another pharmaceutical company that's helping to support us in these regulatory submissions, which is a huge amount of work that often is not very visible to the public, but requires several different functional areas and can span even years in order to get regulatory endorsement or a decision on a product submission.
thank you very much for this. So maybe one question that I would like to ask you, and maybe I will start with Jesus. What has been your most rewarding aspect being part of the COMBACTE project?
Well, that's a good question. I think that there are several important rewards. One is that previously the development of new drugs, all sites that have been participating in the, let's say, pivotal randomized trial, I don't think they have a feeling that they belong to a team. I mean, you participate, you recruit as best as possible, that's it. But here, I think that all sites participating also have the feeling that they were being part of a network, that they could all together provide important information and not only for the pivotal trial, but also, as I explained, for epidemiological data. And that was very rewarding. And I think that was an important point in the success of that study. Also, that being part of the network, we learned from each other. So that means that we share our experiences with other sites. We can discuss about academical interest, about our clinical questions, and also about the aspects of the designs of the studies. And therefore, I think that the best experiences from each site are transferred rapidly to others, and also that feeling of being part of a network is also very rewarding. And finally, by being part of the network, we all have the feeling that we have new opportunities for the future in terms of new collaborations and also new ideas to work together. So everything that happens during the COMBACTE studies for the participating site, I would say that was very rewarding for the investigator in the site.
Anyone else wants to jump on the most rewarding aspect you had being part of this project?
If you ask me for just one, I would say the feeling of being part of a network that provides relevant data.
Yeah, that definitely is also for me the case. So the building of the network and the fact that we have now the network as a separate legal entity that hopefully can continue in the future and can continue to do this work. That, and of course, the pivotal trials that were done in the network, the satellite study and the revisit study.
Same for me.
Yeah, I think for me personally also, this has been very satisfying. Like I said, this is an area that has been a huge need, an unmet need for infectious disease. So it was really, it's been really wonderful to have been part of building such a network, and see that it's now being emulated by other areas geographically, speaking in US and others. But the fact that Europe actually took the lead, and now it will have a legacy in the form of eCrate.
There's still a bit of time if you want to plug your question in the Q&A box. But before that, we have another question, and that will be to both Ale and you, Hassan. Is that what have been the learnings on the company side to conduct the trial in the collaborative way? And since there are a number of companies, and particularly small companies, who are working and developing antibiotics and antibacterial treatment, and they probably are eager to conduct their studies, what are, in view, the impact that the COMBACTE has that can really benefit to them? Or what would you recommend to them?
I'm happy to go first, unless you want to, Hassan.
Okay, yes.
Go ahead, please.
So, the pre-work is such a critical part of conducting a successful trial from my perspective. So we've mentioned before having the right sites in the right place. So with a disease such as antimicrobial resistance, we did a lot of work leveraging the earlier phases of the COMBACTE-CARE collaboration, trying to identify where the areas of highest prevalence were so that we could target our sites there. So I mentioned in my presentation, we had 165 sites across 20 countries. Now, we were able to support something that large because we are a larger pharmaceutical company, but not everyone has those resources. So really investing the time and effort into targeting where is the disease endemic and who are the experts in the area. And we have the fortunate access to this incredible network of investigators who are experienced in research, who are experienced in treating infectious diseases. And so as much as you can to leverage those networks, because that will save you a huge amount of time in the long run, because having to reselect sites while the study's already ongoing, or having to extend timelines because recruitment isn't as you anticipated, there will inevitably be challenges and unanticipated issues that come up in the course of conducting a clinical trial. But to the extent that you can leverage the knowledge of the network and do that pre-work and select the right sites in the right location, I think would be a huge advantage for a large or small company.
I would wholeheartedly agree with what Hallie said. I think for both large as well as small companies, getting in, approaching the network as early as possible is the key, because of course one area is, as Hallie mentioned, is the identifying the places where the patients who have the relevant disease exist, identifying the right sites. But the other thing is, of course, also partnering with these amazing academics who really understand the disease as well as their patients' needs really much better than anyone else. If the companies could really approach the network earlier on at the point of designing the protocol and thinking about the different aspects of the protocol, like eligibility criteria, even thinking about what type of assessments in a clinical trial are feasible and which are not, because a lot of times, you can do too much, and that could actually lead to physicians saying, well, this is a lot of work and we don't really have time and resources to be collecting all this, and plus how is this even helpful for the patient. So I think that type of information a lot of times will come from the academics and from these physicians. So therefore, partnering with them at as early a stage as possible would really bring dividends, because then you can actually design a very efficient clinical trial, as well as have a better idea about which are the appropriate sites. The other thing that we've seen is that when the academic experts are involved, they then become the resource for their colleagues within the region, within individual countries, and that was the idea of the national coordinators that COMBACTE also had introduced and leveraged quite nicely and effectively, and that really helps, because we do know that healthcare systems do vary from one country to the other, and sometimes those could pose some challenges in the conduct of the trial. Having that type of input also is very helpful, and especially for smaller companies who don't really have as much of staff and representation in those countries, having access to that information is also extremely helpful. So those are some of the examples of that.
Thank you very much. Any other points you would like to raise on that from the academic clinician side?
Yeah, I can do. I must say that I've learned a lot also to see the perspective of the, let's say, the pharmaceutical company, because we always look at this from the academic side, but it was also quite interesting to see the perspective from the company side. And indeed, the working with the national coordinators has resulted in the establishment of a eCRADE Clinical Liaison Council, in which the leaders of local and regional, sometimes national clinical trial networks are represented, and with whom we work nowadays in Europe. And that has turned out to be a very, very, let's say, very good decision to start that activity, and that has helped us a lot in executing these clinical trials. Yeah.
We have a last question, and we have only 2 minutes left, but the question is, would it be possible to expand the network to other infectious disease? And the suggestion was on TB. Before one of you reply, I want to mention that under IMI, we do have as well a large project on tuberculosis. And actually in the next coming months as well, we will do something to really report on this important, as we have quite a number of projects addressing the tuberculosis specifically. But I don't know if who would like to take this question for the expansion of the network to tuberculosis?
I'm currently the CEO of eCRADE, so I can answer that question. So I think you gave an excellent answer. I don't think that there is a need for another network in Europe on that disease. We frequently get the question whether we would, let's say, extend our network to, for example, immunological diseases or other diseases. And the answer is certainly not yet. We are still working on a network for acute infectious diseases. There's a lot of work to do in the field of emerging infectious diseases and antimicrobial resistance. It's a very challenging area. These trials are difficult to do, and we will focus on that. It's our expertise. We are experts in these areas, so that's where we can make a difference.
Yeah. And hopefully all of you are still listening to this webinar. You managed to understand really the work and the value of the network that has been created, and importantly, that remain alive through the ECRED, and this is really important. So on this note, we are reaching the end of this webinar, and I really would like to thank you, first of all, our speakers, first for the fantastic presentation and the answer that you gave. I would like as well to thank you, the audience, to be part and being active. And finally, I would like to thank my colleagues from the communication team who has really helped to put together this webinar. And I hope to see you very soon for our next webinar that we will announce in the coming weeks. The first in conversation with will be with another project on RTCure, which is a completely different area, but still interesting to hear about the rheumatology field as well. And on this note, I would like to wish you a nice afternoon, a nice weekend, and we can close this webinar now. Thank you very much to all of you. Goodbye.
Thank you.
Thank you. Bye-bye.